In this video, I'm going to provide a brief introduction to the Lab 6 subsistence exercise and explain a little bit in terms of what we would like you to do and what you need to complete. So in this lab, you're going to be taking some of the materials that you learned in the weeks 8 and 9 content relating to archaeobotanical and zooarchaeological data and applying it. So learning how to interpret these kinds of information. I made an announcement about this already, but just again to correct upon the typo that this is due on November 27th, not on the 26th. So please just pay attention to that. And note that I will always benefit or default towards giving students extra time versus less time if I'm ever incorrect on a date or an assignment. Okay, so for readings, I already mentioned this, that this is the weeks 8 and 9 materials that you'll want to look at, and chapters 12, and it's kind of hidden there behind my comment, but 13 will also provide you with some information. But I've also provided you with some summary information in the document itself. So you can see, I begin with a bit of background information on zooarchaeology. A lot of this just summarizes, again, what is in the weeks 8 and 9 material relating to our analysis of faunal collections, including a brief review of how to calculate MNI and NISP. Okay. I also talk a little bit about taphonomy, which you'll want to look at in terms of this exercise. And then I provide you with a very, very, very short background on archaeobotany, or the study of plant remains, focusing primarily on plant domestication. Now, what's interesting about this lab exercise, or at least I think is interesting, is we're actually going to have you read additional material for this to support your interpretations, and so citation will be part of this exercise as well. So you'll see that we've appended this document by Pearsall 2008 that provides some background on where different kinds of plants were domesticated and when. And this will become clear once you start to read through the scenario why you'd need to know about domesticated versus wild plants. Okay. So for this exercise, we've provided you with the scenario we call the Picard Trapper's Cabin. And so the background is that we're going back to an area that you looked at in the dating lab, which is the Permez Creek region of the Elbow River. And in particular, we're looking at this site located here. So if we look at our key on our map, we see that a cabin is this little square symbol, and our site designation for the Trapper's Cabin is EEP S12. So we're looking at a cabin that's located here overlooking Permez Creek and the Elbow River. And so as you can see here in the background, we talk about how it was occupied based by historical records by Jean-Luc Picard and his Métis wife Marie in a very short period at the turn of the 20th century, so in the early 1900s. And it's been designated as a National Historical Site, and so Parks Canada has done some excavations here to learn a little bit more about early trapping lifestyles and lifeways and subsistence practices in this region at the time. And so our goal is going to be to interpret the archaeobotanical and fauna data that they collected. And that's what we've presented to you here. So in Table 1, we've provided you with a list of the individual specimens that were identified in the 2014 and 2015 excavations. And so in the far column here, you'll see that we use the animal common name. There's an explanation for why we do this above. We have the element, or which bone is represented, which side it comes from of the body. So recalling, as we talked about in lecture, that often animal bones are paired, like long bones are paired in the skeleton. So it's important to know which side of the body it comes from. An indication of its completeness, right? Because not all elements may be complete. Then a an age estimation, then a sex estimation is provided. And in terms of age estimation and sex estimation, sometimes some additional information has been provided in additional notes here, such in the case down here that we have a sex estimation of female, and it says that there's a comment about that this cranium of a deer lacks antlers, which is what they use to designate this individual as female. In other case, we have indications about various taphonomic features that have been identified on the bone. So in this case, we have 2 goat femora, left and a right femur, and both have V-shaped marks. And you can see that in this case, I've kind of given you a bit of a spoiler that V-shaped mark indicates that the bone was cut. Other bones have U-shaped marks. And so again, you have to kind of interpret what this means. And so we have all of this data about all these different kinds of faunal materials that have been recovered from the site itself. And then we're also given some background information and some hypotheses from Parks Canada relating to the botanical materials that were also recovered. So it talks a little bit about the flotation sample, but our problem with this is that while we have this really clear table of faunal remains, our botanical remains have become confused with that from another site. However, luckily, the other site is not a Canadian site, and it's located in the Near East. And we know that the Near East site only contains domesticated plants. So this is where that Pearsall at 2008 encyclopedia entry will become useful, is that you're going to, one of your tasks is to look at the 2 different botanical assemblages indicated here as A and B and try to figure out which one comes from which site. Right? Or not which site, but from which part of the world, right? So assemblage A comes from a site, either our Picard site, our Trapper's Cabin, or it comes from a site in the Near East. Okay. So what are the tasks that ultimately you have to do? Well, first, we're going to go back to table 1 and we're going to use the data provided there to complete table 3. So what is table 3? Well, under taxa, you're going to list each of the specimens. So for example, I mentioned that there's deer and goat, so you're going to put that in here. Then you're going to calculate the number of identified specimens for that taxa. So if we start with goat, we'll say, okay, we have goat, what is the number of identified specimens for goat? So we have to go back to table 1, and you'll recall that NISP is just simply a count of that. So we'll see that we have 1 goat femur, 1 goat femur, so that's 2, 1 goat humerus, that's 3, and a goat cranium, so that's 4. So there we go. I've already given you all your answers. That for goat, again, we go down to table 3, and INSP will put 4, right? Because we have the 2 femora, the 1 humerus, and the cranium. Then you're going to calculate the percentage NISP. So to do that, what you're going to do is figure out what the total NISP is for the site at your very bottom. And then you're going to take 4 and divide it by that total to calculate the frequency, right? And this will just tell you something about, again, the proportional representation of the fauna or the different kinds of fauna within our site. And then we also want you to calculate MNI. So we would do that for here, right? So again, let's go up to the top. So we have for our goat, we have 2 femurs from different sides of the body, from one left and one right, because a goat can only have 1 right femur, so 1 divided by 1 equals 1. So our current MNI is 1. Again, goat can only have 1 left femur, so we only have 1 in this assemblage, so 1 divided by 1 is 1. So now we still have an MNI of 1. We have 1 left humerus, and a goat skeleton will only have 1 left humerus. So again, our MNI is 1 divided by 1 equals 1. And then last, we have 1 goat cranium. I mentioned this in lecture that, of course, mammals only have 1 cranium. So 1 divided by 1 equals 1. And so out of our 4 MNI calculations, we go with the highest, and they're all 1, so that tells us that our MNI for goat is 1. And so we will put that in table 3 down here below. And then you're going to proceed to do that for now every single other taxa that's present. So again, deer, you have some muskrat, beaver, rabbit, cat, etc. So you're going to put all of that in here. Once you've figured out this data, then you can go through and answer the following questions. And I've given you a breakdown of how many marks they're worth each. So you're going to figure out what animals were taken by trappers, right? So summarize what has been found. And we've given you some things to consider in terms of how beaver, for example, have been in decline. So part of the reason why we want to figure out what other animals they were taken is because we know that beaver could not have been the sum of their subsistence practices. Then we have a question relating to age and potentially what were the practices in terms of the kinds of animals. Again, were they conserving populations? Were they culling populations? How are they using them for subsistence? Were they using them only for fur if they were hunting non-fur game species? Why were they hunting them? Was it for the meat? What else was it for? Was it for secondary products? So we want you to think about, again, all the different reasons why people used animals and use that to answer your question. And I'll just note, because I always have students ask me this, you don't have to do research on fur trading for this time period. We want you to answer these questions using only the data we've provided to you. Okay, so don't go and do additional research on fur trapping. We're not looking for the broader perspective of fur trading and of trapping in the early 1900s in Alberta. We just want to know what was happening at the Picard site. Okay. Then we want you to consider whether or not they were keeping domesticated animals, and if they were, what they were being used for. And then finally, we want you to look at how you might separate out some of these different taphonomic factors, right? So what does the difference between V-shape and U-shape marks on bone mean? So, and again, a lot of this information can be pulled from your background. Okay. So that's what you're doing with the faunal data. In terms of the botanical data, we're not going to have you do any additional tables or any different calculations, but what we would like you to do is create a figure. Okay, and so in terms of your figure, if you go down into the instructions, I provide a little bit of information, right? So we want you to create one sort of chart or graph using the botanical data, and we want it to be computer generated. Now, again, some people are going to say, well, I'm not good at Excel. I don't know how to do this. We'd encourage you to try. If it becomes too much of a barrier, please just contact us. I'm sure we can accept bar graphs, for example, that have been generated or have been created using pencil crayons or something and graphing paper or something. But the idea is that we want you to reproduce this table in some sort of graphical form that tells some sort of story about this data. Okay, well, what sort of story can it tell? Well, maybe it's showing the different kind of frequencies of plants between the two assemblages, right? So you might want to create charts that show this to support your answer for what assemblage you think is from the Picard cabin and which one you think is from the site of early domestication in the Near East. Maybe it's about which plant taxa you think were grown in a garden at the Picard site, which ones were collected from the wild. Again, it doesn't matter what the chart is, but really the purpose of this is we wanted you to start to think about how data is represented not just in tables, but also in different kinds of graphs. And so in the dating lab, we had you produce a very specific type of graph, a battleship frequency curves, right? And in this case, we want you to create a graph of your choice, right? So whatever you think is appropriate, that's what we want you to create. And then again, to use it to support your answers for these questions. Okay, so again, so part one is to look at the faunal data. Part two is to look at the botanical data. And then you're just going to, again, typewrite your report and include this in the following sections. So you're going to have an introduction, you'll have your results, which is just simply table 2 and your figure 1. Then you're going to have your discussion, providing your responses to all of the questions. And again, I'll just reinforce this, just like I do in all the labs and on your midterm exams, you need to use the data to support your responses, right? So for example, I'm like, use the MNIs that you've calculated to support your arguments. Because you have a reference for Pearsall, we expect you to use that. And then the last point, this is something new, and it's based on something that Dr. Prince and myself have noticed, is that it's clear that you're collaborating with each other and talking about these problems, which is fine. But in order to avoid academic integrity violations for improper collaboration, because you're supposed to be working on these independently and generating your own assignments, is that if you are talking with your classmates about this, please just simply acknowledge them, right? Say, hey, thanks for the support of, students X, Y, and Z who I discussed this data set with, or something like that. So there's no marks for this. However, failure to do this, and if we see, again, very similar, if not identical responses being provided, it could trigger an academic integrity violation inquiry, which can have very serious consequences overall. Okay, and then I've given you a full reference for that Pearsall article or entry from the Encyclopedia of Archaeology. So hopefully this has answered some of the questions you've had. We've tried to keep this exercise fairly straightforward, but again, don't hesitate to connect with myself or Dr. Prince if you have any questions.